Hello and welcome. This is Ann Holmberg speaking to you again. And we will spend the next 30 minutes talking about correlation and regression analysis. I got a question here whether or not I will share the slides, and the answer is yes. I will share the slides with you after this session. Great. And as always, please use the chat if you have any questions, and I'll try to answer them. Okay, so today's webinar will talk about how to identify and visualize correlated variables. So let's start with that. So we will have a look at the correlation tool to identify correlated variables and also generate lists using search and the set operations tool. I will show you how to generate a correlation network and work with that in a heat map as well. We will compare lists that we've generated in a Venn diagram, and then we will perform regression analysis. And finally, we'll have a look at different options to present your results in scatter and line plots. So we will start with the correlation. So here we will find variables, which could be genes, proteins, and so on, that correlate with one or several selected variables, and we will use the correlation tool for that. And we are here working with Pearson correlation. And here are the steps that we're going to go through. So let's do that. So here we are. I open the software. And I load one of the example data sets, the leukemia data set. I'm sure that you are familiar with a large data set with 132 samples and 22,000 different variables. And we are going to move directly to the variable PCA plot. So now we are looking at the 22,000 genes here. It's a bit crowded, so in order to be able to do anything meaningful here, I need to filter down. So I filter down based on standard deviation of variance. So I focus on the genes here with the highest standard deviation, where we have the most differences in expression between the samples. So I filter down to the 500 genes with the highest standard deviation. And now we will have a look at the variables. I have them here. I put also in the gene symbol, and I sort on gene symbol. And then I'm specifically interested in one gene here, so let's go and look for that. So I want to find CD72. Here it is. Then I will use the mark tool like this to highlight that gene. So that's now marked here in my data set. And if I want to check that it's really CD72, I can label it. And let's change the label to gene symbol like that. Good. And then I can use the correlation mouse tool here. And so here we have a little correlation dialog. And I need to mark my gene here by clicking on it, or if I want to mark several, I can circle them. And then I drag the correlation slider here. So now I'm looking for genes that are correlated with this one. Positively correlated, 63.5% correlation. I can also look for negatively correlated genes. But it's a bit messy, as you can see. There is a lot of other variables in here that I'm not interested in. And as a comment, it's even if the correlated genes are not in the plot, they will be included anyway. So even if you have filtered them away, they will still appear once you do the correlation analysis. So now in order to see what I want to see, I pull the variance slider or standard deviation slider all the way. I filter away everything, and then only the correlated variables are here. And now I can create lists of these. So let's create 2 new lists, and let's call this one positively correlated with CD72. And then I can use the list tool here and just circle these genes like this. And here I want to say negatively correlated with CD72. And then I do the same thing here. I just circle these genes like that. Okay, so that's one way of doing it. So let's just put back everything and clear this correlation tool. In the next exercise, do I have it maximized? No, so let's do that again. We're going to look at another workflow, and here we are going to use the search tool to actually create lists of the genes that we want to work with. And here we're also going to have a look in the Venn diagram of our results and see if there are similarities between the lists. So let's see how we can do that. So here we are again, and this time I want to create a list of the genes that I'm interested in. So now I will use the search tool here in the variables tab, the little magnifying glass here. I want to search on gene symbol, and I want to start with CD3D. So I search for that, and you see that I find one CD3D. Good. And the result from my search is always here in this search list. So I make a copy of that using the copy tool here, and then I rename it to CD3D. Then I would also like to have CD3E, so I search for that, and that is now here, so I make a copy of that, and this is now CD3E. Like that. Okay, so now I've created 2 lists that each only contains 1 gene here, and now I'm going to use these. So let's close this down. So I start with this one. So by ticking this box here all the way to the left, I now select this list of genes. So this is the only active gene right now, and you can see here I only see 1. Let's check that it's the correct one. Yes, it's CD3D. Good. And now I want to use the correlation tool again. I need to click on this gene like that, and that immediately just showed. So let's say I want to stop at around 70%. Here I want to be. And maybe I want to focus on the positive ones like that. So here we are. So then I create a new list like that and use the list and rename it and call that positive correlation with CD3D like that. And then I use the list tool to save this like that. Good. Then I clear this like that, and then I So there are things that you cannot do when you have the correlation tool active. So then you just move away from that. And then I want to take away this now. So then I see everything again. And now I want to show CD3E. Good. So we label that and check that it's the correct one. Yes, it is. Good. And then we want to use the correlation tool again. So we click on this one. And now I find the genes that are positively correlated 70% for CD3E. And you can see CD3D is actually one of them. And then I want to make a list of that. And I can either create an empty list and use the list tool as I just did, or I can also make a copy of the active list. So mark that one and copy. That creates a new list, and this is now positive correlation with CD3E, like that. Good. Then we clear this, like that. And now we are going to have a look at this. So I got a question, so let's see here what it was. Yeah, and the question is, positive correlation now means that if CD3D is up, the positive correlated genes are also upregulated. Yes, that's correct. Because if we have a look here, if we perform the correlation again here, like that, you see that they are all here. And if we also see the, so in the variable PCA plot, it's the same. So if genes are close to each other, they have a similar expression pattern. If they are far away, they have a different expression pattern, the same as in the PCA plot. So the fact that they are close together means that they have a very similar expression. And here they have the opposite expression. So positive correlation and negative correlation here. Okay, now we will move over to the Venn diagram and have a look at the list that we have generated. So here now we would like to compare these two lists, the positive correlated for CD3D and the positively correlated for CD3E. And you can see that 59 of these are the same, and then there are a couple that are unique for each of the genes. That. And in the Venn diagram we can add more. So if I would like maybe to also compare with the first gene that I had a look at, I can do that as well. You can have up to four different lists in the Venn diagram. Let's go back and focus on these two. Okay, so now I would like to know which are these 59 genes. So then I will use the set operations tool over here in the variables tab. And then I want to have the intersection between these two lists. So the intersection between this one and this one. And that creates a new list with the 59 genes. So this is the intersection, of CD3D plus E. And if you would like to know, okay, which are these 11 genes, then or maybe which are all the genes, you do a union, and then you create a new list, and then you have all the 76 genes and so on. And you can also work with the differences like that. So that's quite useful. Okay, any more questions on this before I move on to the next one? The setup window is here. So this is the set operations tool. So in the variables tab, this little icon here opens the set operations. Okay, and now we will have a look at the correlation slider and what we can do with that. So let's go back to the PCA plot here and make sure that we have. So when a PCA plot looks like this is probably because you have marked a list. So in this case now, untick the list and then you will have access to everything again. So let's filter down again to some of the most interesting genes here. So here we are. So here in the method tab, we have networks. As you remember, we have used these when we look for structure among the samples, but we can also use them for the variables. So by dragging the correlation slider here, I look for correlated variables, not specific ones, but every variable that have a 70% correlation with other variables. And then you see that we find different clusters here. So here, for instance, we have one interesting. So let's have a closer look at that one. And I thought we can pull up a synchronized plot and make that a heat map and see what we can do with that. So let's configure the heat map. Let's hierarchically cluster it like that. So if I would like to visualize this cluster now also in the heat map, I can use the mark here and just circle these genes here. Like that. And then I have them here in the heat map as well. And if I now would like to make a list of these genes, I can, of course, use the list tool and circle them as I've done before here. But I can also make a list from the heat map if I like. So I can create an empty list here, a new empty list, and call that correlation cluster or something like that. And okay, now I think let's go back here and do that again, because I think that its mark disappeared. Here we are. And now I would like to use the list tool again and get this cluster here. So let's zoom in a little bit in the heat map first, so it's easier to see. So I zoom in on the variable side like this. Scroll down a bit and I increase the cluster here. So now I would like to grab these genes here. So then I want a cluster that includes these genes, and I think it's this one. So if I click here, I've now pulled in these 33 genes from the heat map. So just teaching you how to also grab interesting variables directly from the heat map. Okay. So let's move on. And we also have a correlation matrix that you can export. So you find that here in File, Export, Variable Covariances, Correlations, like that. You give that a name and you export it as a TXT file, like that. You want to probably include a variable annotation, so I want the gene symbol and I want correlation. Okay. And I have it here. So here you see all your variables here and how they are correlated with each other. So here you can study this to find where you have good correlations. Okay, so now we are going to have, so any more questions on this before we move over to regression analysis? No. So now we're going to have a look at regression analysis. So we can work with linear regression when we have numerical sample annotations like time, dose, age. Then we look for variables that either increase or decrease over the annotation. And we can work with rank regression when you have a sample annotation that is not numerical but with ranked values like low, medium, high, or those type of non-numerical annotations. So let's have a look at that. So we start with linear regression. So then we close down this data set. And the easiest way to do that is from the log tab. Just cross here and then you close everything. And now I will open another data set. Here we are. This is a smaller data set with 12 samples and 50 variables, gene expression. And in here we have the annotation age, which is numerical. And if I color on that, the coloring doesn't really help me to understand the ages here. So let's create a color gradient instead. And I do that by using this little tool here, the brush here. And then it will follow the instructions. Select the first value in the range and press OK. This is my first value. So they must be ordered in the right, correctly in numerical order here. And then we press OK. And then select the last value in the range and press OK. So this is the last value and it's gray. So I would like to have a color gradient maybe from blue to yellow instead. So I changed that to yellow by just double clicking here. And now I press OK. And now I created a color gradient from blue, which are low ages, and yellow, which is the highest age. And so now it's easier to understand what is what here. And looking at the data here, we don't really see a clear pattern. It's a mix of blue and yellow here. But let's now let's open up a new synchronized plot and make that a heat map so we can see what's going on here. And let's configure the heat map and hierarchically cluster it. And also color it on age, like that. Okay. So now we take the statistics dialog here. We select linear regression. We select the annotation that we want to work with, which is age. And then we drag the statistical slider here and see what we can find. And this is not a very strong signal in this data set. So I will stop here at a Q value of 0.3, which is really not good enough. But for the sake of this exercise, we say that it's good enough. And then we find 3 variables, 3 genes here. You can see some kind of pattern, but it's not that fantastically clear. Over here we can see that we have all the yellow samples lining up here and the blue samples over here. So we have found some kind of signal. To make the heat map easier to interpret, we can, when the signal is not very clear, we can manually order the heat map instead to make it easier to interpret. So now it's not unbiased anymore, now it's biased because we have decided how we want to sort it. But that makes it clearer that we have an upregulation over age. Stronger signals for older, higher ages. And we can change the label here to age. So then it's label age, like that. Okay, so that was an example of linear regression. And now we're going to have a look at rank regression. So let's have a look at that. So we can actually work with rank regression also with numerical data. And that means that instead of looking at this number, we just look at the position. So they all have equal weight here. So it's the order of them that we are looking at rather than the specific values. So when we do that, we actually find we find still 3 variables, most likely the same, but the Q value is slightly better. But now we want to do analyze another annotation here, which is the annotation rank. So we have high, low, medium, and for this to work, the ranking must be in the correct order. So we need to move this. So we move high down here. So low, medium, high. That's the correct order. And now we color according to that rank. That, and we color the heat map according to rank as well. And let's see what we get here. So rank regression on the annotation rank. And let's hierarchically cluster here again, like that. Okay, so now we do the same, rank regression on rank, and then we drag the statistical slider to see what we find. And it's even harder to find anything here. So the best we can do here is a Q value of 0.2, and then we find 2 variables here. So the heat map is not, looking at the PCA plot, we see that there is a tendency of high to be over here and low to be here, and medium seems to be all over the place. So let's sort the heat map instead of on the annotation rank and see if it's easier to understand what we have found, like that. Yeah, and then we see again that we have an upregulation for rank and a downregulation for upregulation for high, a downregulation for low, and medium is a bit of a mixed signal here. Yep. Yeah, any questions so far on this? Or everything is crystal clear? So then we are going to spend the last few minutes here talking a bit on how you can visualize your results here. So let's say that we would like to visualize some of these variables in a scatter plot. So let's change here to the method tab, scatter plot. So scatter sample we have here. And here I would like to have the sample annotation age on the x-axis with a numerical scale. And then I would like to select the variables manually here. So I go here, and then I select the list of the 3 variables that I forgot to save. That was stupid, so let's recreate them. Linear regression on age, and I find my 3 variables here. Yep, so here I have them. So these are the 3 variables that we found when we did the linear regression on age. So by just selecting that list, I get them here. By clicking on the y-axis, I can configure and say that I would like to show the symbol here. So here we see that they are all they have an increasing signal over age. We see that the correlation coefficient is not fantastic, but we still see the tendency. If I would like to label, I can just use the label tool. If I would like to color, I can do that as well, maybe on treatment, for instance. And if I only want to see one variable, for instance, then I just pick one variable from the list here, like that. And then there are more options in the scatter plot. So now I will load the other data set again, which contains a bit more data, becomes a bit more interesting. So now we can also change this to a scatter plot. And maybe I would like to show one variable on each axis. So let's do that. So let's just pick one gene and another gene, like that. And again, maybe we would like to show the gene symbol. So you can do that easily as well. So you can show one gene and one variable on each axis. And then we can, of course, color this according to something, leukemia subtype, for instance, like that. Then we can show line plots. So let's change this to a line plot. And then we have all the active samples on the x-axis. And then we can pick again what we want to show. So let's pick, for instance, one of the lists that we created initially here, the positively correlated genes for CD72. And then you can see here that they have a high and good correlation. And here are the negatively correlated ones. So this can also be a very nice plot if you want to have a look at several variables over the samples. And we have more plots. We can, in the scatter plot, we can also say that we would like to have a, we go back to scatter here, scatter sample. Maybe we would like to have a sample annotation on the x-axis. So let's say, like to have leukemia subtype, and then I select which variables to show. So that's another way you can do that. And you can also make a 2D PCA plot. So if we go here, we can say I would like to have PCA component 1 here, and I would like to have PCA component 2 here, like that. So let's filter down a little bit to make this more interesting. Like that. So here we have a 2D PCA plot. And you can also be in the variable mode here, scatter variable, and then you have samples on the axis. So then we select an individual sample here from sample. So let's go down and pick, we have some stronger signals. Here we have one, and then I would like also to select the sample here. And here we have another one. And then maybe we would like to color. So now we are showing the variables here. So we have one sample on each axis, and maybe we would like to color the variables according to how they are expressed for instance, this leukemia subtype, like that. So then you see that we have an upregulation here and a downregulation here using the color tool. Okay, so that was what I had planned to show you today. So, any questions on that? So I got a question here. How did you export the correlated values to Excel in such a table? I guess you mean the correlation matrix. So let's have a look at that again. So here, File, Export, Variable covariance correlations. Then you just give it a name. Save it. Tell which sample annotation you would like to show. If you want covariance or correlation, and then you export it to a TXT file. That was the question. Yeah, good. Anything else? So then I would just like to remind you, if you haven't already attended these webinars, in your case, maybe the two last ones here could be of interest if you haven't already attended them. More information on how to work with RNA-seq, single cell and proteomics data, and how to work with gene set enrichment analysis for pathway analysis. So this is everything that we're going to do now before Christmas, and then we will start again beginning of next year. And I guess that all of you are already have Qlucore installed. If you don't, you can download a free trial from our homepage. And I will send you the slides so that you have them and you can try to repeat this yourself. So by that, I would like to thank you very much for your time today, and don't hesitate to contact me if you have any questions. Just send me an email. Thank you so much and have a nice day. And there is a little evaluation form when you exit this webinar. So if you have time to give some feedback, that's very appreciated. Thank you so much. Bye-bye. Have a nice evening.